Okay, so we've talked about T lymphocytes, and they're involved in well, the T helper cells and cytotoxic T cells. Cytotoxic T cells kill cells that are infected with viruses, and T cells facilitate that process. Now, we're going to talk about another kind of lymphocytes here, which are called B cells, and they look down the microscope very similar to T cells. They look exactly the same as T cells, but they're different. And these are involved in making antibodies, which you will have heard of, like like GCSE. The name for antibodies is really called immunoglobulins. And right, so let's have a look at the structure of an immunoglobulin molecule here. Right, there's 4 peptide chains. You can see there's 2 big ones, those big ones here. 2 heavy chains, and the 2 ones, these are called the light chains. And they're actually held together by disulfide bridges, if you remember what they are. And this bit of the molecule it doesn't vary, and it's not involved in binding the antigen. What the external bit, this other bit here does, it will bind. So here is an antigen. Okay, so that may be like a bit of a bacteria, or a virus, or a little bit of a multicellular pathogen or something, but it will bind. So this is the antigen here, and it gets bound. Now, the difference of this antigen and when we talk about T cells, this is not presented by another cell. It's just floating around in the blood or in the tissue fluid, free. It's not in the major histocompatibility molecule like was the case with the T cells. So it's just floating around as a bit of bacteria or a virus which has infected a person. Okay. Now, immunoglobulins, or Igs, can exist in, most of them are soluble in the blood. So, there's people are trying to get a test for the coronavirus to see who's had it, test for immunoglobulins in someone's blood, immunoglobulins that will bind to the coronavirus, and that's a good indication that they've been infected with it. There's another kind of immunoglobulin which is cell bound, and that is bound to the surface of the B cell, right, just like that one there. Now, this is a very similar situation to the Tcell receptors. The immunoglobulin, there are millions of different types. So, for example, that one there is, this one I've drawn here may bind a bit of one particular bacteria, but there'll be lots and lots of different types of immunoglobulin. Each one will bind something slightly different. Okay, and now how do they all arise, even though there's only one gene for an immunoglobulin, or there's one set of genes anyway? Well, it's the same procedures with the T-cell receptor. You start off with a B cell progenitor in the embryo. You make a load of different, okay, from that you get millions of different sorts of B cell. Each one's got a different immunoglobulin on the surface, and that's how do you end up with lots of different immunoglobulins when there's only one set of genes? Well, again, the DNA is cut and spliced together in different ways, so you end up with lots and lots, millions of different B cells, each one with a different immunoglobulin on the surface. Then in the embryo, that undergoes some kind of selection, because all the ones that react with self, they all get eliminated in the embryo. So the only ones that left over, these recognize foreign antigens. And again, there's all kinds of, there's B cell which will recognize antigens which haven't even come into existence yet. So they cope with every eventuality. Okay, so what happens here is, let's say you've got this B cell, and it's got it can bind that red antigen. Okay, so the red antigen will bind there, to the surface of the B cell, and that will activate the B cell. Now, in order for this B cell to be fully activated, it needs T helper cells to produce these interleukins, interleukin 4 and interleukin 5, and they help it. They help the maturation of the B cell. Now, the B cell will then start dividing like mad. Will divide over and over, lots and lots. So one cell can give rise to millions of other cells by dividing and dividing. But they turn into they can differentiate then into two different kinds of cells. One lot turn into the plasma cell. Now, if you look at the plasma cells, they've got lots of rough endoplasmic reticulum. Okay, and the reason for that is they make lots of immunoglobulin, which is a protein. That's why you need lots of rough endoplasmic reticulum. And the immunoglobulin that they make is this one. Okay, it's the soluble version of the one which is on the surface of the B cell. So it was just producing one kind of immunoglobulin, but it's the one that will react with the bacteria or the virus or whatever that the body has detected in the blood. So they chuck out loads and loads of this immunoglobulin, and that helps in fight infection. We'll talk about how it does that. But also they produce a kind of cell called a B memory cell. And what that does is, as the name suggests, well, it's got the correct immunoglobulin on the surface of it, the one that will just hang around in the lymph glands for the rest of your life. And if you ever come into contact with that antigen again, then they're all ready to very quickly divide, turn into plasma cells, and mount a very rapid immunoglobulin response that can help fight the infection. Okay, so that's what B cells do, and that explains, acquired immunity, how you become immune to things. Okay, now let's just talk about how immunoglobulins, how do they end up killing bacteria? Well, first of all, they can produce agglutination of bacteria. Because they've got two binding sites per molecule, you've got one there and one there, then you can get this sort of thing where they all kind of, they cross-react, and that will have another, so it forms these great big aggregations of the antigen all together with the antibodies, and this can then be phagocytosed quite easily. Also, if you have a bacteria, so here's a bacteria, okay, and you've got an immunoglobulin stuck to it, well, this region, this is the other end of the antibody here, it's called the FC region, and that has, the macrophages have receptors for that. The macrophages recognize that, and they phagocytosis, and the phagocytosis is more efficient when the bacteria is coated with antibodies.